During meiosis, gametes or sex cells are created. These gametes are haploid, not diploid cells, which means they only have one set of chromosomes. Interphase is not a part of meiosis, instead it is where the cell grows. During gap 1 or G1, its cell membrane and nuclear envelope expand. During the S phase or synthesis phase, the cell duplicates its DNA and its centrosome. Instead of one single strand of DNA, there are two known as sister chromatids. And during G2, the cell grows even larger. During prophase, the chromatin in the nucleus condenses. Synapsis aligns the homologous chromosomes or homologs side by side, and they condense further into chromosomes. Crossing over begins, in which genetic material between non-sister chromatids is exchanged in the homologs. The point of crossing over is the chiasma. This results in recombinant chromatids increasing genetic variation, which is also caused by independent assortment in which homolog pairs are randomly paired. Then the nuclear envelope breaks down. During metaphase, the homologs line up on the equatorial plate. In anaphase, the homologous chromosomes are pulled to opposite poles by the spindle fibers of the centrioles. These spindle fibers are made from kinetochore microtubules.
In telophase 1, a nuclear envelope is forming and the cleavage furrow forms. Homologous chromosomes pairs complete migration to the two poles as a result of the spindle. Then the haploid set of chromosomes are at each pole, with each chromosome still having chromatids. In cytokinesis, both of the nuclei are formed. The cell pulls itself apart, resulting in two daughter cells. This happens as the same as telophase 1 occurs. In interkinesis, it's a period of rest that cells of some species enter during meiosis 1 and 2. It's the resting part, per se. In prophase 2, the centromeres move to the polar regions and arrange spindle fibers for the second meioic division. Nuclear envelopes disappears, they break apart, the chromosomes condense, and the centrioles move apart for the spindles to start forming.
Now we move on to metaphase 2. In metaphase 2, the centromeres of the paired chromatids line up across the equatorial plates of each cell. The difference between this and metaphase 1 is that pairs of chromosomes are aligned in metaphase 1, while pairs of chromatids are aligned in metaphase 2. In anaphase 2, the chromatids separate and become chromosomes in their own right, moving to opposite poles. Due to crossing over and independent assortment, each new cell will have a different genetic makeup. In this phase, the number of chromosomes are cut in half. The sister chromatids are formed, leaving half of what would be found in anaphase 1. In telophase 2, the chromosomes gather into the nuclei and the cells divide. The spindles once again disappear and the nuclei reappear. By the end, we will be left with 4 new cells. These cells are still considered to be haploid. In cytokinesis, each of the 4 cells results in a nucleus with a haploid number of chromosomes. The plasma membrane pinches in to stabilize the cell and deposit new cell wall materials. This completely separates the cells that were once together.